All right, hello again. We're going to pick up where we left off. So, we finished in class talking about the physiology of the autorhythmic cells, or those cells within the heart that spontaneously depolarize that ultimately lead to a contraction, right? And those autorhythmic cells, they're connected to the other cells within the cardiac muscle, which are the contractile cells. Remember, 99% of those muscle cells are contractile, only 1% are autorhythmic, and they're connected via these gap junctions. So today we're going to talk about the physiology of those contractile cells. As always, to kick us off, a couple of memes. We have this one here. This was a favorite of us students 5 years ago. It's true. I got kids screaming in the background here. I'm at home, sorry. And then remember that I said there are multiple names for the left AV valve, and one of those was the mitral valve. So this isn't exactly a joke, but here you can see it gets that name because it looks like the mitre or the hat that sits upon a bishop's head. I don't know if you can see it. I don't, but... Alright, so where are we going to go today? Well, we're going to start out with the contraction of a heart, and that's going to include those myocardial cell contractions. Myocardial, myo just meaning muscle, cardial, heart. So we're talking about the muscle cells that contract here. And then we're going to talk about this phenomenon called excitation-contraction coupling. I'll try to illustrate that for you as best I can by doing a live drawing. All right, we'll begin with myocardial cell contraction. And if you haven't already stopped and printed off the worksheet for today, worksheet 3, go ahead and do that. I will be collecting this when we get back. So this will be due in class when I see you again. Okay? So fill this out while you're working through. It's pretty straightforward. All right, if we look at this image here on the right, it's showing us the layers of the heart. So this innermost layer is called the endocardium, and then next to that we have the myocardium. Myo again meaning muscle. Okay? So this layer, not to kick a dead horse here, but you can imagine this question is going to show up on an exam, but this layer is comprised of 99% contractile cells and 1% autorhythmic. Now these contractile cells in there, they're highly vascularized, so they have this nice red color. They need that vascularization to deliver oxygen adequately for this thing that is continuously beating, right? We talked about adequate perfusion. It's going to contain many mitochondria, which is going to allow it to have epic, we say, aerobic capability. So think back to last semester when we're talking about aerobic capability. We're talking about these cells that are going to be very fatigue resistant. They're kind of the ultimate slow oxidative fibers that are just going to continue to keep beating with a adequate supply of oxygen. Now, remember I said they're kind of strange and that they're a combination of skeletal and smooth muscle, really. So the similarities to skeletal muscle are that they contain the myosin and actin contractile components. Okay? They're also arranged into sarcomeres, that contractile unit that shortens. The differences are they are much smaller than skeletal muscle fibers. Remember skeletal muscle cells, that's also called a fiber, they span the entire length of the muscle. This isn't the case here. There are many cells linked together, and they are branched, and they have around 1 to 2 nuclei per cell, so they can be multinucleated. That smaller cell and branching offers a very large surface area. And a larger surface area is going to allow for maximal diffusion of things we need like oxygen, glucose. It's going to provide easy entry of those into that cardiac muscle. When we look at cardiac contractile cells, they look like this. So we can see here that some of them are branched, and then you see these lines here separating the individual cells. We call these intercalated discs. And this is where these contractile cells are connected to one another. Okay? These intercalated discs, if we zoom in, it would look something like this top image here, which is an actual image of contractile cells. And now these dark vertical lines, this is where these branched individual cardiac contractile cells are connected, intercalated discs. Okay? So let's see what goes on at an intercalated disc. Okay, there are two kinds of membrane junctions that we can see at these intercalated discs, or where these cells, cardiac cells, contractile cells, connect to one another. So if we look at this image here, we can see the end of one cell, and that is about up next to another cell here. And here are those two types of membrane junctions. And the first one is important for the physical connection of these cells that tightly holds them together. And that is the desmosome. Desmosomes you might have referred to last semester as tight junctions that hold cells tightly together. And they work just like a rivet on your jeans, right? That hold two pieces of fabric together like a pop rivet. So you can see there's this protein here on one end and on the inner membrane of the other cell, and then they're connected via these proteins in between to kind of sew these two cells together physically. Now, the other type of membrane junction that's going to be important for these cells is going to be those gap junctions we talked about last semester. So gap junctions are going to allow action potentials to spread from one cell to other connected cells, right? So this is going to be very important, and it's going to allow the cardiac tissue to act as a functional syncytia. Remember we talked about the functional syncytium last time, and that is that all those cells, because they're connected via those gap junctions, are going to contract as a single unit. And that is how the heart contracts as one single unit due to these gap junctions, which form a functional syncytia. Okay? So again, two important types of membrane junctions here. One that holds those contractile cells together, and another that allows for communication between them. All right, so here we have two cell membranes up against each other. So I'll just label this 1 and 2. And these are two separate cells. They could be two contractile cells. They could be two autorhythmic cells. Remember, they're connected via gap junctions as well. Or maybe it's an autorhythmic cell that's up against a contractile cell. They are also connected via gap junctions. Well, what is a gap junction? Well, it consists of these little subunits, 6 on each membrane. So you get 1, 2, 3, sorry, I'm using my mouse pad, 4, 5, 6, okay? And those make a little tunnel. And each one of these is called a connexin, okay? Connexin, look at this image here, IN. And when 6 connexins come together, they form a connexon. Right? So a connexon molecule is 6 connexin subunits. Okay? And then we have a connexon, that molecule, on each side of the membrane. So over on the other membrane, we would have 6 connexin subunits that form one more connexon. And when these two come together, they form a tunnel through the membrane. And we can see that here. Here you have a connexon, right? And the other side you'd have a connexon as well. And when those two adjacent membranes come together, that completes a whole tunnel in which ions can flow through, right? So those connexin molecules meet across the membranes, and they provide a passageway for ions. Specifically, ones we're interested in are going to be sodium and calcium, for example. Then I have this little saying down here in Latin, unus pro omnibus, omnis pro uno. And I don't know if any of you know what that means, but it means all for one and one for all. Right? So if you've ever heard of the Three Musketeers, that was their saying, right? And that's because gap junctions allow for that functional syncytia. That's if one of our autorhythmic cells depolarizes and contracts, all of the cells within the muscle tissue of the heart will, at least the contractile cells. So it's all for one or one for all. If one contracts, they all do. Unlike skeletal muscle, we do not have recruitments of fibers here, right? So if you're going to lift something really light, you wouldn't necessarily need to engage all of those skeletal muscle cells. Here in the heart, we can't control that, because if one autorhythmic cell depolarizes, it spreads through every cell, and every cell contracts because it's a functional syncytia. So all for one, one for all. And just to start putting this all together, remember we have this collection of autorhythmic cells at the S A node, okay, the sinoatrial node, and that's where this whole contraction should kick off if everything's working normally within the heart, okay? And then via gap junctions, it's going to spread through all the other autorhythmic cells and through this conduction pathway, which is connected to all these other contractile cells within the atria and ventricles via gap junctions. So we get contraction of the entire heart as one, albeit at slightly different times due to the delay that occurs at the AV node, right? And you can see that well on this image here. The atria contract first, followed by the ventricles. That is because we need to eject that blood completely from the atria before we start contracting the ventricles. And I should say that spontaneous depolarization at the SA node that kicks everything off, again, is going to be due to those funny channels, or that slow drift to threshold, that slow depolarization that occurs due to those funny channels that open upon hyperpolarization that we reviewed last time. All right, I drew a similar image on the board in class last time. Okay, so if we look, let's just say we're looking at the SA node here. So this is your SA node. And remember, that's a collection of autorhythmic cells. So let's say that's what these are here. These are clustering of autorhythmic cells within the SA node, right? And they're abut up to these adjacent cells, and they're connected via gap junctions, those autorhythmic cells to the contractile cells. And then all these contractile cells are connected together by gap junctions at those intercalated discs where they're physically connected to each other by those desmosomes. Now we need two events to occur if we want these cardiac cells, contractile cells, to contract. So they're going to be stimulated by the conduction system or these autorhythmic cells, right? We're going to get spontaneously depolarizing cells, the autorhythmic cells, and that current is going to spread via gap junctions to the contractile cells. So the first event we need is propagation of the action potential at the sarcolemma. Okay? And remember, the sarcolemma is the outside of the muscle cell, right? Okay. And the second thing we need is we need contraction of the sarcomere in the cardiac cells. So remember, we said these cardiac cells have a contractile unit, the sarcomere, just like skeletal muscle. Well, we need that to shorten. So in order to get cardiac cell contraction to occur, we need propagation of action potential along the sarcolemma and then contraction of the sarcomere. And we call these events together, we call it excitation, contraction, coupling, or ECC. So that's what we're going to work through next. We need those two events to occur. We need the cell to be excited that leads to contraction of the sarcomere. So excitation, contraction, coupling. Let's start by looking at the propagation of the action potential along the sarcolemma. Okay, the first event we need to occur in order to get this contractile cell to shorten or contract is propagation of the action potential at the sarcolemma. Okay, sarcolemma, remember, is just the sheath that the muscle cell is covered in. Okay. So if we zoom in on the sarcolemma, we'll see some proteins that we're familiar with. Okay, so the first thing we're going to have that's not shown there would be a sodium-potassium pump, and that's going to help establish a resting membrane potential. Okay, so notice we do have a resting membrane potential now, unlike our autorhythmic cells, which were not able to maintain one due to those funny channels. So here our resting membrane potential is negative 90 millivolts. Now some other channels you'll recognize are going to be a fast voltage gated sodium channel. We're going to have a slow voltage gated calcium channel, and then our regular voltage gated potassium channels. Along the sarcolemma, we're also going to have potassium leak channels, sodium leak channels, and the previously mentioned sodium-potassium pump. All right, let's start by looking at the changes within membrane potential that occur in order to give rise to a contraction. So here we have our standard membrane potential over time. And remember, this is telling us what's happening inside that cell relative to the outside in regards to the charge. Okay, so the first thing that we see here When action potential reaches the sarcolemma from an autorhythmic cell, is we see a depolarization. Okay, so that, remember, that is becoming less negative. That depolarization is followed by a plateau. So this is kind of unique before. Usually we just start going up and then come back down. Here we have an elongated plateau that slowly starts to repolarize. And then that's followed by that sharp repolarization ultimately. Okay. Now we're going to look at which of the channels and proteins are involved to give us this characteristic depolarization. All right, here is going to be the contractile cell action potential. Typically I would draw this on the board, but I'm going to give it a go drawing it on my tablet and then insert that on the next slide for you. All right, so just as we looked at the changes in potential that occur across the membrane last time with the autorhythmic cells, now we're going to look at the contractile cells. So this is very similar. And remember, these changes or fluxes in current that we are looking at here, they're what is changing in the inside of the cell relative to the outside. Sometimes we're becoming more positive compared to the outside, sometimes more negative. So whereas last time we were getting these changes in autorhythmic cells due to funny channels that were opening upon hyperpolarization, now we're going to be looking at the changes that happen in contractile cells when that depolarization from the autorhythmic cells reaches those contractile cells via gap junctions. Okay, so the first thing we're going to have then are just our axes like we had last time. All right, so we got a y-axis and an x-axis. Of course, this is going to be membrane potential, and we measure that in millivolts. And then down here we have time, and we measure that in milliseconds. Okay. Just some things we can plot out here. This will be plus 30 at the top of our axis here. Let's say our threshold, that is going to be at negative 70 for these contractile cells. And we're going to go as low as negative 90, right, when we're polarized. Okay. So now let's add some of our proteins or channels to this figure. And I'll try to color code everything here. So let's do it this way. Put this off to the side up here. Let's go ahead and say that blue, everything we put on this figure in blue, that's going to be a calcium related event. So Ca2 plus. Let's say that green, is going to be potassium related, and let's say that red then is going to be sodium. Cool. Okay, so the first thing we're going to start out with then is a little bit of green, and we're actually not going to start at the first step here. It makes a little more sense just to start at the fourth so we see what's going on. So here we are. We're going to be down at, when we are polarized, down at negative 90. Okay. And this is going to be our resting membrane potential here, around negative 90. Okay. It's going to be a little more negative than we're used to seeing in things like neurons, for example, because these potassium channels here are leaky. So I'm going to call this K and little sub L. So we'll put a little 4 above this. And because we have potassium leaking out, right, remember that's a positively charged ion, so when that's leaving, we become more negative. So we're down here at negative 90, a little more negative than usual. And let's go up here and start it like that. So this is step 4. And 4, we're going to have our potassium channels, K+, leaky. These are going to be open. And that's where we get this resting membrane potential of around negative 90 millivolts. But then we have that incoming depolarization from those autorhythmic cells. Okay. Now, this is going to involve a sodium flux. So we're going to have a voltage-gated sodium channel that is going to open now. So we're going to see this rapid depolarization. We're going to go all the way up to positive 30. Okay, and if that was step 4, now we're going to start, and we're going to call this just arbitrarily step 0. So we'll go over here, and let's write out what's going on at step 0. Well, this is now voltage-gated sodium channel that is opening. You get that incoming depolarization from autorhythmic cells that triggers the opening of these voltage-gated sodium channels. Remember, they're voltage-gated, so that depolarization is going to cause them to open up. Okay, now we're back to our voltage-gated sodium channels acting in more of a classical fashion, right? That's opening upon depolarization from that incoming signal. Not like with our funny channels when they're hyperpolarized, they open. So what we see with this is a rapid depolarization all the way up to positive 30 millivolts. Okay, and why does that happen? Well, the sodium rushes into that contractile cell. So we have a lot of positive charge moving into that cell now relative to the outside. Okay, now we're going to involve another potassium channel. And we're going to see this small repolarization here, and we're going to call this step 1 now accordingly. And this is going to be K plus T. Oh. So that's a small TO there if you can't read it. And that means transient outward. Okay. And I am going to go back here real quickly and just label this. Since we put it everywhere else, this will be Na plus. Back to potassium here. So at step 1, we have this transient outward potassium channel. So that is a voltage-gated K plus transient outward. That is going to open. Now again, when we say transient, that means kind of short-lived. So it's only open for a short period of time. And you can remember outward, just that's the flux direction that the potassium is going to be leaving the cell. Okay. When that leaves the cell, we get a little bit more negative, right? So we start repolarizing a little bit, and that's where we see this decline. Okay. Now the other event that you can note that happens here simultaneously, and I'll try to color code this correctly, will be those voltage-gated sodium channels from before are going to close. So voltage-gated Na plus close. And because those close, then we stop having positively charged sodium ions entering the cell, and that's how we start to decline here and return back to polarization. All right, next we have a calcium channel. It's the first time we've looked at those here today. And this is going to be event 2. And here, we're going to have just this plateau. Okay. So this is event 2 now. Involves calcium. You see a 2 plus. So here we have these voltage-gated L-type calcium channels open. Remember that L stands for long. They're open a long time. And we also, at this step, have our voltage-gated potassium transient start to close. So if we look at these two events here, to kind of explain why we see this plateau. Remember, a plateau is just a flattish area here, and that's what we see here. So if we think about this, it makes sense, right? We have our potassium channels that are open. So potassium is, because it's been concentrated within that cell, via the sodium potassium pump, it is leaving when we add a little channel for it there in the membrane. So that positive charge is leaving, we start hyperpolarizing. Not hyperpolarizing, repolarizing. Okay? But then we have some calcium channels open. And calcium then is going to move into the cell. So that's a positively charged ion moving into the cell. So you have these two things that are fighting each other here, right? You have potassium leaving, so becoming more negative, but calcium entering, so the cell's becoming more positive. And they kind of cancel each other out, and we get this plateau, where it's just kind of flat for a while. Okay? So that's how we have that plateau. And then we have our third event here, which, again, is going to be related to potassium. Let's see this. Depolarization. So this, I'm sorry, repolarization. And this is going to be step 3. At step 3, we have these voltage-gated L-type calcium channels. Well, they're now closed. And because they close, the positive charge with those calcium ions stops entering the cell. Right? And if that stops, then we become more negative, especially when we have these now voltage-gated potassium channels. I just call these ordinary, the ones we're familiar with. They open. Okay, so this is another type of voltage-gated potassium channel. They open, which means a bunch of potassium is now rapidly leaving that cell. So I'm going to put K plus out. And if that's leaving that contractile cell, now we become negative again. We repolarize. And we're allowed to do that because those voltage-gated calcium channels, where calcium was entering the cell, are now closed. Okay. Last thing I'm going to add on here, I forgot to add it, our threshold at -70. And that's just when that depolarization from the autorhythmic cell allows us to hit -70. That's when we get this action potential. Okay. And with these two events, this allows for a repolarization. And we approach -90 again. The very last thing I want to point out here, let's do a new color. How about some orange? Is that if we were to plot a contraction of a contractile cell on this same figure, we would have different axes, but it would look something like this. So that plateau corresponds kind of to the length of a contraction. Why is that? Because in between here, that gives us an absolute refractory period. And we'll talk about the significance of that next. Basically, we cannot get a contraction during that plateau phase no matter what we do. And that is going to be very important for the contraction of the heart. Let's take a look at that. All right, so thinking about that refractory period that we just finished up with in that video. Can't obviously do a think, pair, share because we're not in class, but you can think about this individually. Why would this refractory period be important for the heart, especially concerning the contraction of the heart? Why would we need a refractory period? What does a refractory period ensure? Well, it ensures that another contraction can't happen right away, right? And this is going to be really important for the heart because the cardiac muscle cannot exhibit tetany. Remember, that's a sustained contraction. So unlike skeletal muscle, cardiac cells have this long refractory period where the cell cannot fire a new impulse during that refractory period. So we see that this corresponds to that plateau phase, which leads to a refractory period of about 250 milliseconds. During that time, the heart cannot contract again. It's an absolute refractory period. And this is very important because this ensures that there's alternate periods of contraction and relaxation, which are essential for pumping blood in the heart. Imagine if you had a sustained contraction or tetany without any or a very short absolute refractory period. Well, then you could have a contraction right away after the heart has contracted, maybe not finished contracting all the way, or finished relaxing and filling with blood all the way. So this allows the heart cells to contract and relax before it can be stimulated again. If you had tetany within the heart, you would not be pumping blood, the brain would not get oxygen, and you would die. Okay? So this absolute refractory period makes sustained or tetanic contraction impossible, which is necessary for us to continually pump blood throughout the body. Now you may be asking yourself, what exactly gives rise to this long absolute refractory period? And it's something you're familiar with previously. Remember, we had our voltage-gated sodium channels. We just saw some within the video that we watched that lead to that rapid depolarization. Remember, they have the different states, right? They have those different gates, inactivation gates, activation gates, that give rise to the three different states: closed but capable of opening, open, and then closed and not capable of opening. Because of the slow release of the inactivation gate to clear that channel, right? So that's so slow that we have this refractory period. Well, it's those exact voltage-gated sodium channel qualities that give us that absolute refractory period here following the depolarization within our contractile cell. So it's the same qualities, these voltage-gated sodium channel qualities, that lead to one-way propagation within the neurons, right? The signal can only flow one way down the axon in a neuron because the channels behind it are closed for a period due to that inactivation gate. Okay, well, it's the same thing. Those inactivation gates are closed for longer within these contractile cells that give us that really long absolute refractory period. Here's an example of that absolute refractory period within the neuron, and you can see how that leads to one-way propagation of signal down the axon, right? So it moves in one direction because the gates behind that signal are closing and then are remaining closed due to that inactivation gate for a small period of time. Now, it's a small period of time within our neurons, right? It's 1 millisecond absolute refractory period, whereas now we're talking about a 250 millisecond absolute refractory period in these cardiac contractile cells. So those inactivation gates are closed much longer. Okay, so we finished with the first event of our cardiac cell stimulation, the propagation of action potential at the sarcolemma. Now we need to look at the second part, the contraction, or the actual physical shortening of that sarcomere within the cardiac cells, right? So we're talking about, remember, excitation, contraction, coupling. So we've talked about excitation. Now we need to talk about the contraction component. All right, so now we're turning to how that propagation of action potential along the surface of the muscle cell is linked to the physical shortening of the cell. Okay? And the linking of these two events is called excitation, contraction, coupling. We're linking the excitation at the cell surface to the physical shortening of the cell, the contraction. So excitation, contraction, coupling. Now to kick us off, I need to do a little bit of review on what's going on at the surface of the cardiac muscle cell. Let me get my laser here. Okay. So remember the outside portion of that contractile cell is called the sarcolemma. And then notice it has these little holes, right? And these are the T-tubules that dip in through the surface of that muscle cell to allow us to access the individual myofibrils inside, the sarcomeres that actually physically shorten and contract. Okay? And notice these are surrounded by the sarcoplasmic reticulum. And remember that sarcoplasmic reticulum, or SR, is where we store calcium that is going to be released to lead to a contraction. Okay. Again, I am going to do another drawing on my tablet. This typically would have been done on the board. Let's take a look at how action potential leads to contraction, or how incoming depolarization leads to the physical shortening of the contractile cell. And the first thing I'm going to make a note of here that we need to remember is that the contraction is going to be initiated by extracellular calcium. Initiated by extracellular calcium. So this is calcium that's coming from outside of that contractile cell. So let's draw the sarcolemma. So this is going to be a zoomed in version of the surface of that contractile cell. So this is if we were looking at it from the side. Here's our bilayer. Now, this is our sarcolemma, right? But this little dip here is going to be our T-tubule. So remember, that's how we access the internal part of that cell and all those myofilaments that are comprised of the sarcomeres, the contractile units. Some other things we need to draw in here would be the sarcoplasmic reticulum. And remember, the sarcoplasmic reticulum surrounds all these myofibrils and sarcomeres that allow for calcium release. So in here we have a whole bunch of calcium within the sarcoplasmic reticulum. Okay. All right, and then there are some channels that are going to help us with our concentration gradient of calcium. So the first of those I'll draw in the membrane here on the sarcolemma. Call this channel 1. And this is going to be our sodium-potassium pump. So remember, with 3 sodium inside the cell, 2 potassium. That does 2 things for us. It keeps the concentration of sodium inside the cell low, on the potassium inside the cell high. So it establishes a gradient that we can use in the next step. And this next one here we'll call 2 is the appropriately named sodium calcium exchanger, NCX. And it does just that. We're exchanging sodium for calcium. So we established this gradient by concentrating sodium outside the cell here, just with our sodium-potassium pump that we just drew. Okay. So sodium is going to want to move into this cell where the concentration is low. We'll draw that. So 3 sodium move into the cell, which can then power, so down its concentration gradient, which can then power the movement of calcium up its concentration gradient. And it's pumped outside of the cell. And in this way, we can keep the calcium concentration within the cell low. Now, there's one other way in which we move calcium. The third way. And this is just a straight up primary active transport, so ATPase pump. It uses ATP directly to move calcium against its gradient and concentrate it outside of the cell. Okay? So this is primary active transport. This one is relying on that sodium concentration, so this would be secondary active transport, okay. And this also would be primary active. Just turned 2 the other day. Okay. So, just to review here, sodium-potassium pump concentrates sodium outside of the cell. That sodium wants to move down its gradient, and then moves through this sodium-calcium exchanger down its gradient, which allows us to power the movement of calcium against its gradient concentrated outside the cell. The other thing we can do, we can use this calcium ATPase pump, use ATP directly to move calcium against its gradient and concentrate it outside the cell. So basically here what we're creating is this large extracellular gradient of calcium outside the cell. Remember, this is important because this contraction is going to be initiated by extracellular calcium. Okay, let's draw some other things here. So let's just say we have an incoming depolarization. So we have an incoming action potential. This can be from other contractile cells or maybe an autorhythmic cell. Doesn't matter. We have incoming action potential. Now, within these T tubules, that is going to reach a specific protein here. And the one we're interested in is going to be those L-type calcium channels. So just go back and look at the membrane potential drawing we just did. It is the L-type calcium channel that kind of starts this, allows for calcium to slowly enter the cell, contractile cell, for a long time, which gives us that plateau phase. So action potential reaches this L-type calcium channel, and we're going to see an influx of calcium. So this is that extracellular source of calcium. That's a voltage-gated channel. It's going to be triggered to open up. Calcium is going to move in. I'll add voltage here just so we're clear. Now, where does calcium go upon entering the cell? Well, upon that sarcoplasmic reticulum, remember, we have a certain receptor, and that is called the ranitidine receptor. R-Y-R. Ranitidine receptor. So calcium goes and binds the ranitidine receptor, and that allows for release of calcium from the sarcoplasmic reticulum. So this is called that calcium leading to the release of more calcium is called calcium-induced calcium release. So we have incoming calcium that induces the release of more calcium from the sarcoplasmic reticulum. And it works out that about 20% of the calcium source is extracellular, so coming in here, but it leads to more calcium release from the sarcoplasmic reticulum. 80% of the calcium that is ultimately released that helps with the contraction. Okay? So that is calcium-induced calcium release, a term you should be familiar with. But it is extracellular calcium that kicks off the whole thing. Okay, so now we've released calcium. And what is that going to do? Well, remember, this is releasing it right next to those myofibrils, those contractile units, the sarcomeres. Remember, that's comprised of thick and thin filaments. Thick filaments here, sarcomeres. And it is this calcium here that leads to shortening of these contractile units. Remember, we need calcium to bind, to move the tropomyosin out of the way to allow for contraction. Okay? So these are our myofilaments here. And when those sarcomeres shorten, the entire contractile cell shortens. Now, At the end of the contraction, we need to get calcium back into this sarcoplasmic reticulum. So here we have the circa pump. If you're not familiar, that stands for sarcoplasmic endoplasmic reticulum, calcium, ATPase. So with that ATPase, we know we're going to directly use ATP to move calcium back into the sarcoplasmic reticulum using ATP. Okay, so just to work through the entire process one more time from start to finish, we have incoming action potential, incoming depolarization. That depolarization activates our voltage-gated L-type calcium channel. That opens up, and we've been concentrating calcium outside of the cell via these mechanisms here, right? That allows for a calcium concentration gradient. Calcium wants to move into the cell. When it does, it binds to the ranitidine receptor. Binding of the ranitidine receptor causes more calcium release from the sarcoplasmic reticulum. Okay? This is calcium-induced, calcium released. 20% incoming calcium from extracellular sources leads to 80% release from sarcoplasmic reticulum. Calcium comes along, binds the troponin, pulls the tropomyosin out of the way, and allows for interaction of contractile units or contractile components, and we get physical shortening of the cell, or contraction. Okay? At the end, we get that calcium back into the sarcoplasmic reticulum via the circa pump. Okay? So now we have a detailed look at how calcium enters the sarcoplasm, or that fluid within the contractile cell. Remember, we have two different sources. Some is coming from extracellular sources, and the majority is coming from that sarcoplasmic reticulum. And we call that calcium-induced calcium release, where that extracellular calcium, it's binding sarcoplasmic, or the ranitidine receptor on the sarcoplasmic reticulum, and leading to even more calcium release. So we have a bunch of calcium now floating within the cell. What does it do? Well, just as like in skeleton muscle, it's going to bind to that troponin molecule. And when it does that, it pulls the tropomyosin out of the way, exposing the binding sites for the myosin heads. And we initiate what we call cross-bridge cycling, or contraction. So when that cross-bridge forms, we get the power stroke. That myosin head pulls on the actin, and then it resets. When do we stop this contraction? Well, over time, calcium levels decrease, leading to relaxation. And when do calcium levels decrease? Well, that would be when the action potential stops, when that cell stops receiving incoming depolarization. When that happens, the channels close, and the pumps then, like the circa pumps, move it back into the sarcoplasmic reticulum or back out of the cell via some of those other mechanisms, primary and secondary active transport we talked about in the previous video. And if you need a little refresher on the mechanics we just talked about, here we are. So we've released calcium into the sarcoplasm. Some's come from extracellular sources, majority has come from the sarcoplasmic reticulum. But it's floating around here, and then it can bind to the troponin molecule. When it binds troponin, there's a conformational change, and it pulls this rope-like structure, the tropomyosin, out of the way, exposing the binding site for this myosin head. Okay? And then that power stroke can begin. Triggers a power stroke that pulls that thin filament inward during a contraction. And the entire cell then physically shortens. Eventually, action potential stops, and we start seeing the closing of the associated calcium channels. We have continuous reuptake of calcium from the sarcoplasm back into the sarcoplasmic reticulum by the circa pumps. So that's continually happening, as well as the transport of calcium back, out of the cell We have things like the sodium-calcium exchanger or the primary calcium ATPase pump. All right, and finally a very nice summary slide that puts everything we've talked about today together. So first off, we have action potential that occurs, and that can be incoming from another contractile cell or from an autorhythmic cell. Anyway, that signal travels down the T tubules. Upon entering the T tubules, that allows for the opening of those L-type calcium channels, and we get a small amount of calcium coming in through the extracellular fluid. This initiates calcium-induced calcium release, where we get a large amount of calcium released from the sarcoplasmic reticulum. Right? These both contribute to the amount within the contractile cell calcium. Remember, it's about 20% versus 80%. But either way, we have an increase in intracellular calcium. That calcium binds troponin, pulling tropomyosin out of the way, exposing the myosin binding site. We get cross-bridge cycling with those power strokes. And we get physical shortening of our sarcomere that we call the contraction. And as your activity, you will complete the questions on your worksheet that are follow-up to lecture regarding contractility of these cardiac cells. An example on your worksheet is asking you to consider calcium channel blockers, or CCBs. How would this class of drug affect heart rate or contractile force? And can you think of a condition where this would be necessary? Again, this worksheet will be collected when I see you again, and also find your list of objectives for Lecture 3 on Canvas as well. All right, we'll see you in class.